Welcome to my coronavirus classroom. I'm Janessa Jacobs, and this is another ridiculous review from my living room. This time we are reviewing the skeletal and muscular systems, sometimes collectively called the musculoskeletal system. So let's just start with a general stuff review. What's the most important stuff in anything? Well, always the anatomy and the physiology. So if we start with bones, as far as the anatomy of bone goes, we have bone tissue, which is a connective tissue, and the cells that we have are going to start out as these osteoprogenitor cells. So all of our connective tissues are related because they start from the mesenchymal stem cell line. Some of those are going to differentiate and become osteoprogenitor cells. Those are going to be able to become the cells that actively secrete osteoid and growing bone, called osteoblasts. So if you remember, osteoblasts build bone, so they lay down osteoid and growing bone. Osteoblasts are going to surround themselves in osteoid, and that's going to solidify, and they'll become entrapped in a lacuna and become an osteocyte. Osteocytes are the mature bone cells that maintain bone matrix. So they are able to take up, say, excess calcium in your blood and deposit that in osteoid. So those are our mature cells. Osteoprogenitor cells can also become these other cells called bone lining cells. And they support our cells in our membranes. They're found in the membranes, the endosteum and the periosteum, and support our other cell types. The other important type of cell that we find that opposes the action of osteoblasts and osteocytes, it breaks down bone matrix, are our osteoclasts. And our osteoclasts are more closely related to macrophages, which are blood cells, than they are to our bone cells. And these are the cells that break down osteoid. So when we need to access our body's reserves of calcium, our osteoclasts will break down osteoid and release that to the blood. Okay, so those are probably the most important things we need to worry about as far as cells go. As far as other things in bone anatomy, well, you have to know all your bones. 206 bones in the body you need to be able to identify the important markings that you're required to know from your lab guide. So make sure you go over that. But thinking about physiology, so that's the anatomy, the cells, the tissue. I guess that's not all of it. What other tissue type do we find in the skeletal system? So we have osseous tissue, or bone, which comes in two forms, really, compact bone and spongy bone, that have all of those cell types we just talked about. Okay, well, what do we find between our bones? What tissue type? Cartilage, right? So we have three types of cartilage. The most abundant is hyaline, and we find that covering the ends of our long bones. It forms our costal cartilages, forms the bulk of your nose. That is going to actually form the basis of our bone formation in the fetal skeleton, which is where we're going next for a physiological process. But let's just get through the anatomy. So hyaline cartilage is pretty easy to identify because our matrix is pretty amorphous. That's the distinguishing feature. In fibrocartilage and elastic cartilage, you can really see protein fibers in a way that you just don't in hyaline cartilage. So in fibrocartilage, where do we find that? Intervertebral discs, pubic symphysis, and the menisci of the knee. And in fibrocartilage, we have, what's different about it is that we have, or what's easily recognizable about it, is that you find this really nice arrangement of our collagen fibers that are packed very tightly. You'll also see your chondrocytes entrapped in a lacuna, and the lacuna are pretty easy to identify. So your chondrocytes will be in there, and you'll have this nice arrangement of these really thick collagen protein fibers, and that does what for fibrocartilage? It gives it great tensile strength. So fibrocartilage can resist compressive forces. So, yep, that's that for fibrocartilage. And then our last is elastic cartilage. We find that in areas that we need to have flexibility and bounce back. So your epiglottis, your external ear. Okay, those are our tissues. Now in our anatomy, of course, there's joints and things. So forget not about that. But let's think about physiology. So how do our bones grow? Ossification. So ossification is the process of forming bone tissue, and this is going to happen by two methods depending on what kind of bone you are. So we have intramembranous ossification, which is going to happen within like a membrane that looks, a connective tissue membrane that looks like the bone. So this was my membrane for the frontal bone, believe it or not, which is your bone right here. So flat bones of your skull formed by intramembranous. And the membrane is this mesenchymal mold. So bone starts as connective tissue in utero, and so our intramembranous bones are going to start as like this mesenchymal mold, and then we'll start seeing the appearance of these ossification centers. So some of those cells, those stem cells, are going to differentiate and become our osteoprogenitor cells, and our osteoclasts, and our osteoblasts, and we'll start differentiating, and you'll see that we get all these ossification centers, and as that bone grows more, next you'll see what we call the formation of woven bone. You can kind of see it starts to look lamellar, and then it starts to look spongy, and then we lay down compact bone outside it and we get our bone. That's not the one that I really want you to know in great detail. You should have a general idea about what intramembranous ossification is and the bones that form using it. But the one you need to be able to know in great detail, or be able to put in some kind of steps, is how most of our bones, it's really easy to identify. So all our long bones, it's very easy to see the zones of growth, and this is going to happen by what we call endochondral ossification. Endo is within, chondral is within a cartilage mold. So our chondrocytes are the mature cells, our chondroblasts are the immature cells that lay down our extracellular matrix, because cartilage is also connective tissue. So for endochondral ossification, let's think about this. So we've got this diaphysis, which is the long shaft of your bone in the middle, and we've got these two epiphyses that we're going to have to solidify also. And then all throughout development, we're going to want cartilage to be present in some place where we can lengthen out but have a good, long, strong diaphysis. So I guess I should have drawn this over here. This is where we're getting to at the end of ossification, is kind of this goal where we have a cartilage growth plate here within this what was totally cartilage mold. So we start in utero with a hyaline cartilage model of the bone. So it looks something like the bone is going to look, and you'll see just this connective tissue, mesenchymal membrane first, and then that will form into our hyaline cartilage. And then around our hyaline cartilage, we'll see the development of this bone collar. And this is all starting from the same stem cell type. So some of these like cells in here are differentiating and becoming chondroblasts that are laying down the extracellular matrix of hyaline cartilage. Well, some of them can also differentiate and become osteoprogenitor cells, which can lay down bone. And so we'll see the formation of a bone collar around the diaphysis. So it's really important to get this long center of the bone formed and solidified. And in that, then, the next kind of thing that we'll see is this periosteal bud is going to invade. So the periosteal bud invades. And in the center here, we'll see these chondrocytes start dying off because of the high calcium in the area. And so we'll see this space form. This will be our primary ossification center. So we'll get this space emptying out. This periosteal bud is invading. What does the periosteal bud contain? The arteries, veins, lymphatics, so all the vessels and nerves that serve the bone, because bone is living tissue. It's a living organ. It's served by all of it. So the periosteal bud is going to contain that and come in here. And what's cool is that as it comes through this bone collar, it's going to bring with it our bone cell types. So they can come into this primary ossification center and start filling in these empty spaces with bone. So we'll start seeing that now that we ossify here and we get hard. And so then we still have our cartilage in here and our cartilage in here. And then we see secondary ossification centers form here in the epiphyses. And more blood vessels come in to serve. And now we'll see our spongy bone is going to form here. We'll lay down compact bone on the outside, so that by the end of endochondral ossification, what we've done is totally ossified all that hyaline cartilage that was in the diaphysis via the primary ossification center. And then after our secondary ossification centers form, we'll form a bunch of spongy bone in here, which is important. Why? What happens in spongy bone? It contains red bone marrow, which is the site of hematopoiesis. So, okay, that's going to be important. Have to have blood supply there. But we're going to lay down some compact bone and ossify in our secondary ossification center. So our epiphyses will ossify. And now, at the end of endochondral ossification, all that will remain as far as cartilage goes is the cartilage here in the proximal and distal epiphyseal plates, and then the hyaline cartilage that is also found as the articular cartilage. So just make sure you can put that all in steps and in your own words. How we start with a hyaline cartilage mold, and so that hyaline cartilage mold will have the chondrocytes die off, our primary ossification center forms, our bone collar forms, primary ossification center forms. Then as that happens, it's an ossification center, so bone is going to start forming here. Our periosteal bud is going to bring all the blood vessels and nerves that serve. It's bringing in our bone cells, and ultimately is going to result in this calcification, ossification of the diaphysis. Then our secondary ossification centers form, and they get their blood supply. Compact bone is laid down, we ossify it, and at the end, then this is where we are from birth through adolescence and puberty. So what's happening during puberty is you're having your last big growth spurt, and then you're going to solidify and ossify these epiphyseal plates. So what does that look like? I apologize if you feel like this review is more confusing than whatever. You can always go back and watch the regular lectures. It's just also good to have things reviewed a million ways and think about it. When you're done, I challenge you to turn this off and write out in step form 1, 2, 3, 4, 5, the steps of endochondral ossification. And then go look at your book. Don't look back at the video because it was kind of all over the place. It's ridiculous. It's a ridiculous review from my living room. Go look at your book and see how your steps compare to what they outline for endochondral ossification. If you've got it all down, you're good to go for the test. If you don't, then you need to study it again. And you can either watch the silly review again and put it all back together, or you can watch the lecture that is still available by just, clicking on it in your former announcements. Okay, so that's ossification. That's how the bone forms. But how does the bone grow? And how do we, so when you're born, when you're, little, your bone may be this long, but when you're a teenager, it needs to grow this long. So how are we growing, and then how are we completely ossifying? So that happens through postnatal bone growth, and that is where we have those different zones of cartilage that we can find here. So remember, at the end of endochondral ossification, cartilage only remains at the epiphyseal plates and as articular cartilage. So let's look at this again. Here's our diaphysis. And it was ossified all throughout endochondral ossification. Here's our distal epiphysis. Here's our proximal epiphysis. Those were also ossified by our secondary ossification centers during endochondral ossification. And now what we have left is hyaline articular cartilage covering the end of our proximal and distal epiphyses. And we have hyaline cartilage at the epiphyseal plate, or the growth plate. And if we look, growth is happening in opposite directions on opposite ends of this bone, right? Because in here, this is bone. This has been ossified. It was cartilage, and it got ossified. And we have cartilage right here. And so what's going on? Well, we're growing from the inside out. What are you talking about, lady? Does this even make sense? It does. Let's look at it. So we're going to look at what's happening on both epiphyses. And it's opposite what's happening on each epiphysis. So if we look at the zone of growth or development that's closest to the epiphysis, that's going to be opposite on each end of the diaphysis, right? Right, because the epiphysis is here, the epiphysis is here. So let's put this as number, we're just going to call it like naught. Because this is like when we're numbering our zones of growth, the first is, number one is our proliferation zone. So our first zone that we'll talk about right here, closest to the epiphysis, we'll call it zone naught. This is our resting zone. And here, the chondrocytes are relatively inactive. They're resting, okay? So that's closest to the epiphysis. All right, our next zone is where we're going to be growing or dividing by mitosis so that we have more chondrocytes in number. So our next zone, zone 1, is our proliferation zone. And what's happening here in zone 1 is our chondrocytes divide by mitosis. So what does that mean? They increase in number. So now I have more chondrocytes in here. Okay? And since they're kind of in a it's kind of cool. Since they're in a semi-solid ground substance, they're going to form. This is happening in a lacuna. So you could see like one nucleus, and then it'll undergo mitosis, and then you'll see two, and then they're going to push away from each other and get bigger in the next zone, the hypertrophic zone. So our next zone in, closer to the diaphysis, is our hypertrophic zone, and this is our zone of growth. So our chondrocytes are going to increase in size and push apart from each other in these big lacuna. So this is our hypertrophic zone. And what we could say here is that our chondrocytes increase in size. So now we're getting bigger and pushing away. So if we look here, this is happening on opposite ends. So we're growing. Well, hold on, we are not to the whole growth part yet because we're still talking about cartilage. It'll make more sense in our next layer. Okay, well, the next two. So chondrocytes now are growing, getting bigger here. They do not like very, very like hypercalcemic environments. Well, bone is a hypercalcemic environment. This is where we store our calcium. So in this third zone, the zone of calcification, that's what's going to happen. We're going to accumulate a lot of calcium and kill off the chondrocytes. And that is going to leave big open spaces for our next thing, which is to ossify. So in our calcification zone, calcification, what happens here? We could say the extracellular matrix calcifies. This kills off chondrocytes. And this is going to leave big open spaces. So now we've got these big open spaces here that can now be filled with osseous tissue. So now in our zone here, our fourth zone, which is closest to the diaphysis, which is bone, we're going to make this what was cartilage bone. So our zone closest to the diaphysis is becoming bone. So do you see how we're growing from the inside out? The bone is happening here, or the calcification is happening here. So what happens after this ossifies is that we move out a little bit on each end. Okay? So our last zone is ossification. Ossification. And we could say our bone cells fill the empty spaces and ossify the bone, which is pretty awesome. So again, this is something that's important, and I would be able to either number out or describe in my own words. So after you have described in your own words endochondral ossification and compared it to the answer in your book, take a break, have a cup of coffee or some tea, then come back and describe postnatal bone growth in your own words, and then look at your book and see how it matches. Could you number and place the zones in both zones appropriately? Can you see how this actually works in the body? If I were you, I sure would try. Okay, quick question. What's interstitial growth? When did we even talk about that? We did, barely. But your book talks about it too. Interstitial growth is growth in length, like we were just talking about. That's growth in length. What's appositional growth? Appositional growth is growth in width. So just be aware of that. The periosteum is the membrane that surrounds the outside of the bone. It's perichondrium when it starts in cartilage. But the periosteum is going to be how we're growing appositionally. It's laying down bone on the outside. What cells on the inside, on the endosteum, might be hollowing out the medullary cavity so that it's growing as the periosteum is laying bone on the outside? Let me ask that question again. On the inside of the bone, our medullary cavity has to be hollowed out and grow appositionally as well as while we're laying bone on the outside. So what cells on the inside hollow out the medullary cavity? Osteoclasts, okay? And then of course we're gonna have to lay down more bone, and you build up and break down and build up and break down. It's always going back and forth between osteoclasts, osteocytes, osteoblasts. Just during development it's osteoblasts and osteoclasts. Bouncing back and forth. But then when we need to later access our stores of calcium, our osteoclasts will break that down. And then next time you have a lot of calcium and we can lay it back down, our osteocytes are going to pull it out and lay it down in bone matrix. So it's always this balance between those cells. Just be aware of that. So that's appositional growth is happening in the periosteum and the endosteum for that matter. And then interstitial growth is the growth that we're talking about that's happening at the epiphyseal plates. All right, so as far as the skeleton goes, yeah, you have skeletons. So use your skeletons to name all the bones that you have to be able to identify. And we've been doing bones of the day this entire time, so there's not really an excuse for not knowing a whole bunch of them already. All right, the next chapter, joints. Okay, we can classify our joints based on anatomy, and we can classify our joints based on physiology. Yeah, there's an entire lecture video already posted this. I don't really, what do I want to say? I would say study joints, try and figure out how, what's a good way for you to figure out, I don't know, which ones move where. Or think about while you're studying your skeleton, could you name every joint or the type of joint between all the bones? So for example, you have to know the bones in the skull and you have to know the cranium pretty well. What bone is this? It's your frontal bone. What bones are these? Parietal bones. What bone is this? Occipital bone. Okay, you have to know the sutures. So you have to know the names of the specific sutures, but you also have to know what type of joints they are now. So when you're studying, test yourself the whole way down the skeleton. So this is our what suture? Coronal suture. Okay, sutures. They have to have an anatomical classification, what they're made of, and a functional classification, how much movement they allow or what they do. What's found between sutures? Little fibers that get ossified. This is a fibrous joint. So that's its structural classification, is it's fibrous. Specifically, it's a suture. And is it synarthritic, amphiarthritic, or diarthritic? It's synarthritic. It should allow no movement. Okay, so study all of your joints that way. Why not? Here, my jaw. How much movement do I have? What type of joint is it? Okay, this is my mandible, and my mandibular ramus leads to my mandibular condyle, which fits into the mandibular fossa of my temporal bone. Those are all things that you have to know, and you should know by now. So, or maybe not, well, yeah, by now, this is your review day. So, okay, what type of joint is that? What type of movement does it allow? Functionally, it's actually a diarthrosis. It's freely movable. The only joints that are freely movable are functional, or by structural classification, synovial joints. Okay, so that's how I would review your joints, and that's the most important stuff for your review of the skeletal system. Muscular system. Oh my goodness, another really complex thing. But let's just do a quick review. So how do we contract a muscle? A lower motor neuron is going to send down an action potential that is going to stimulate this muscle. Okay, so how? Let's talk about it. What the heck are all of the parts that matter for contraction of a skeletal muscle? Our lower motor neuron, this is the synapse. This is our synaptic cleft. This is our motor end plate. And our motor end plate is specialized why? If we look at an entire muscle fiber, which could be as long as my muscle that it resides in, they can be real long. Okay, so my entire muscle fiber, what makes this one spot different? Why is this the motor end plate? It's these receptors here. What is going to open this receptor? A chemical or a voltage or something mechanical? A chemical, right? A neurotransmitter, specifically acetylcholine. So acetylcholine is being stored right here. It's going to be, it will bind our ligand-gated receptors. These are ligand-gated channels. Acetylcholine receptor is a ligand-gated channel. When acetylcholine binds, it's a cation channel. What happens when we open a cation channel? Cations move, both move. Sodium moves in, potassium moves out. We have a net influx of sodium. That's going to create our end plate potential. Okay, so this was our neuromuscular junction. A neuron is here. Our muscle is here. So this is my neuromuscular junction. And at the neuron, I had an action potential that came down my lower motor neuron. I had the consecutive opening and closing of my voltage-gated sodium channels, followed by the consecutive opening and slower closing of my voltage-gated potassium channels. So that when I get down here, the depolarizing swing opens up my voltage-gated calcium channels. Calcium comes in, fuses with the proteins that are holding these neurotransmitter vesicles in place. We can release neurotransmitter by exocytosis. The neurotransmitter is acetylcholine. It crosses the synaptic cleft, binds to our acetylcholine-gated receptors. Those are ligand-gated cation channels that will allow for a net influx of sodium that causes the end plate potential. Now, if you thought you didn't need to know action potentials after the nervous system, you were wrong. That's why we talked about action potentials then, because I wanted you to understand how neurons work, which use action potentials, and we still have to understand action potentials for our muscle. So now, down the length of our sarcolemma and T-tubules, remember, we've got to bring this action potential deep into the muscle fiber to surround our myofibrils. So that contain our myofilaments for contraction. So we've got to get this action potential deep down in here. This end plate potential is going to result in a net influx of sodium. And as long as we get enough sodium here to get us to our threshold for the voltage-gated sodium channel, then we will conduct our action potential in both directions away from the motor end plate, down the sarcolemma, and deep into the T-tubules. What's the threshold for our voltage-gated channels? Negative 55 millivolts. That negative 55 millivolts, our voltage-gated channels open. Sodium is going to dump into positive 30 millivolts. Then the voltage-gated sodium channel is going to inactivate. Our voltage-gated potassium channel will take over, and we'll see our potassium exiting we're not, the resting membrane potential of our muscles is -90, so we're not going to hyperpolarize. We're not going to worry about any more of that, other than knowing that's what's going to happen. Potassium is going to exit and brings us back to rest. What we need to talk about is what happens as this action potential continues. So this positive 30 millivolts opens up our next voltage-gated sodium channel, so we rush into positive 30 millivolts. Opens up the next voltage-gated sodium channel, so we open up to positive 30 millivolts. Now, when we get into the T-tubules, this depolarizing swing is going to open up voltage-gated calcium channels here in what? This is another anatomy thing that you have to know. There are triads. On each side of a T-tubule, we have two. On each side we have one, but a total of two terminal cisternae, or terminal cisterns. And what these are is these enlarged regions of sarcoplasmic reticulum that are storing a lot of intracellular calcium. So this is specifically our endoplasmic reticulum. And it's got a whole bunch of calcium stored inside the cell. So as our action potential comes down this sarcolemma and T-tubule, the depolarizing swing is going to open voltage-gated calcium channels in our terminal cisterns of the endoplasmic reticulum. Calcium is going to flood the sarcoplasm, and then we'll be able to start the events of contraction. So this was excitation. This that I just described was excitation-contraction coupling, and it occurs down the sarcolemma and T-tubules. The last event is what happens when, now that we've released this calcium, we can bind to our contractile proteins and change the conformation so we can actually start shortening and contracting our muscles. So there are three parts. What happens at the neuromuscular junction? Sure, it's got lots of parts. The second part is what happens in excitation-contraction coupling. Sure, it's got lots of parts. The last thing is what happens in contraction, or the events of the cross-bridge cycle. So, a sarcomere spans from one Z-disc to one Z-disc. Z-disc are these proteins that are anchoring our thin filaments, and our thin filaments are pointing in to the center of the sarcomere. Our thin filaments have three proteins. Actin is the contractile protein. And then we've got this tropomyosin-troponin complex that's blocking the myosin head binding site on actin and relaxed muscle. So this is our tropomyosin-troponin complex. Okay? Those are the proteins in actin. The other part of our sarcomere, in the middle we have the M line. These are proteins that are anchoring our thick filaments. Our thick filaments have what? Myosin. And coming off of that, we've got on it these little spring-like proteins, titin, or some books call it connectin. Either way, what happens as we shorten this, we compress this, so that when we relax, we don't need anything to bounce back to our resting length. Okay, but the contractile protein is myosin, and it's got these myosin heads that are going to be able to reach up and grab onto the myosin head binding site on actin once this regulatory complex is moved. How do we remove the regulatory complex? Calcium binds to troponin. It shifts the shape of the tropomyosin complex so that now we can expose the myosin head binding site on actin. Myosin reaches up, grabs the myosin head binding site, and begins the cross-bridge cycle. The first thing that happens is a cross-bridge forms. And when the cross-bridge forms, myosin reaches up and grabs onto the myosin head binding site on actin. The second thing that happens is the power stroke. What happens is myosin enters its low energy conformation and just does that. So it was here and it goes like this, but it was grabbing onto actin up here, so the actin comes closer together. The next thing that's going to happen is we bind ATP and we let go. But not all of them are letting go. Some of them are in a different, they're trading positions in their cross-bridge cycle. So some of them are holding on, some are letting go. The one we're talking about is now going to let go. It's going to do this because it binds ATP, so now it's not going to bind actin with such affinity. It binds that ATP, hydrolyzes it, and resets. So we're going to reset or cock back to the high energy conformation. When we started So what we do is we do our power stroke, we let go, we hydrolyze energy, come back to the position that we started in, and we reset. And now, as long as we've got calcium present and we're still, our myosin head binding sites are still exposed, we can reach up, form a cross bridge, do the power stroke, let go, bind ATP, hydrolyze it, reset to the high energy conformation, reach up, and go over and over and over again, as long as calcium and ATP are present. So, yeah, that's the cross bridge cycle. Probably the most important, the complex part of muscles. That's not necessarily true because you have a ton of muscles to memorize. So hopefully you've been working on your muscles of the day. So for everything listed in your lab guide, you need to know where to find it and what it does. I would memorize my origins and insertions while I was at it because it may or may not get me some bonus points at a later date and time. I think that's the most important stuff. So that's my general review for you. Here's some questions. Which of the following is not a cranial bone? A, the ethmoid bone. B, the sphenoid bone. C, the vomer. D, none of the above are cranial bones. E, all of the above are cranial bones. Yeah, the answer is the vomer. The vomer is not a cranial bone. Cranial bones touch the brain somewhere. And our ethmoid bone and sphenoid bone both touch the brain. The vomer is found inferior to the ethmoid bone, and with the ethmoid bone actually forms the perpendicular plate of your nasal cavity. So if you look into your skull, you'll see there's, like, right in the middle of the nasal cavity, a perpendicular plate. That is the ethmoid bone. And, well, the perpendicular plate of the ethmoid bone meets up with the vomer, and that's what makes that nasal septum. Okay, so which statements regarding articulations is not true? A, articulations range in motion from freely mobile to immobile. B, articulations occur between a bone and another bone, cartilage, or a tooth. C, all articulations contain some type of cartilage. D, all of the above are true statements. E, A and B only are true statements. All right, the answer is C. All articulations contain some type of cartilage. The question is, which statements regarding articulations is not true? This is not true. Fibrous joints don't have cartilage in them. Fibrous joints have fibers. So yeah, that's just not true. So sure, cartilaginous joints do, yes, and synovial joints have cartilage, yeah, supporting and reinforcing, well, you've got ligaments and all of that, but there's definitely articular cartilage present, but our fibrous joints do not. So some people might be saying, okay, I chose E, why is this not true? Well, this is true. A and B only are true statements, but it's not answering the question. The question is asking what statement regarding articulations is not true. And while this might be a true statement, it's not the answer. So you might hate that, and you probably do hate that. It's a nitty-gritty testing thing, but this is what matters when you're sitting in your, like, future entrance exam, like your NCLEX if you want to be a nurse, or your, what is it, PSATs if you want to be a pharmacist, or your MCATs. It's these types. This is what differentiates those who get into medical school and those who don't. Those who don't get into medical school choose this true statement but don't answer the question. So I'm just trying to teach you how to do whatever you want, including answer really nasty test questions. Okay, the following tissue is subject to subconscious control. A, smooth muscle. B, cardiac muscle. C, skeletal muscle. D, all of the above. E, A and B only. The answer this time is A and B only. Smooth muscle and cardiac muscle are controlled by, well, cardiac muscle sets its own pace, which is super cool. But we can adjust it with the autonomic nervous system. Smooth muscle, for sure, controlled by the autonomic nervous system. Okay. In order to receive the signal to contract, each skeletal muscle must be served by an A, artery, B, vein, C, nerve, D, all of the above, E, A and B only. The answer is nerve. You might be like, yeah, but I chose all of the above because we need all of the above. Okay, here's another question. In order to contract, each muscle must be served by All of the above. So see the difference in the question? Yes, in order to contract, a muscle has to have everything. In order to receive the signal to contract, the signal's coming from a nerve, okay? From a neuron or a nerve, okay? All right. The following contractile proteins are found in thin filaments. A, actin. B, tropomyosin. C, troponin. D, all of the above. E, A and B only. Yeah, actin. Actin is the only contractile protein. Now, some of you definitely said all of the above. Sure, they're all found in a thin filament, but not all of them are involved in contraction. Only actin is. These are the regulatory proteins that block the action of myosin on actin in a relaxed muscle. All right. See this. If I asked it a different way, the following proteins are found in thin filaments. It would be all of the above. These are important testing strategies we're learning here, people. Okay. Which of the following is not paired? Which is not paired? Maxilla, mandible, zygomatic bone, all of the above, A and B only. That means it doesn't have two. Only the mandible doesn't have two. This could have given you a hint here. Maxilla, one is just an A, the E on the end, because there's, yeah, making it all fancy in Latin or Greek. Maxillae. So you do have two maxillae and two zygomatic bones. All right, amphiarthritic joints can be. So amphiarthritic is its functional classification, structural classification, fibrous, cartilaginous, synovial, all of the above, A and B only. Yeah, this time it's A and B only. Fibrous joints and cartilaginous joints can both be found in areas where they allow some movement, amphiarthritic. So let's think of an example of each. The reason that synovial doesn't work is because synovial joints are considered diarthritic. All synovial joints are considered diarthritic, even if they have varying ranges of motion. I know that might drive you crazy, drives me crazy, but I still have to teach it because this is still true. So synovial joints are freely movable. Fibrous and cartilaginous joints are either synarthritic or amphiarthritic. So for our fibrous joints, sutures are synarthritic. They allow no movement. Gomphoses also allow no movement. Those would be synarthritic. But our syndesmoses, which are the little, like we can find in our interosseous membranes, where we have just little bands of fibers, those would be an example. A syndesmosis is an example of a fibrous joint that's amphiarthritic. Okay, now let's think of our cartilaginous joints. This is where we have cartilage between our two bones, and we have our, if we have hyaline cartilage between the bones, what do we call those? Synchondroses. So our synchondroses we find like between true rib 1 and the manubrium of the sternum. Synarthritic, no movement. The epiphyseal plate that we just talked about is a temporary synchondrosis that occurs during growth. Also synarthritic. So then it must be our other type of cartilaginous joints, our symphyses. Our symphyses, remember we said our intervertebral discs are pads of fibrocartilage. Those are amphiarthritic. They allow a little bit of motion, and the more you move your vertebral column, the more motion you get. So our symphyses would be an example. Your pubic symphysis, it can allow a little bit of, like, for expansion. It shouldn't be moving a whole bunch. I would call the pubic symphysis should mostly be synarthritic, but our intervertebral discs are definitely amphiarthritic. And yeah, hopefully that was a great review of the joints for you without being too confusing. If it was too confusing, my apologies. It'll hopefully make sense soon. If not, you can always post a question in the discussion board any old time. Okay, tendons and ligaments can attach to bones using tuberosities, trochanters, epicondyles, all of the above, A and B only. All of the above. So yeah, like your deltoid tuberosity is where your deltoid attaches. Your greater and lesser trochanters are on your femur. All sorts of muscles attach in there. They're awesome to hold that leg in place. Hold your thigh in place. Oh, amazing. Epicondyles, like yeah, your medial epicondyle, attachment point for some of our supporting ligaments in synovial joints and things. So, all right, next, when thinking of the pelvic girdle, the joint gives up blank for blank. It gives up flexibility for stability. So your humerus, your glenohumeral joint, gives up stability for flexibility. Strength was just a distractor. When thinking of joints, we're looking at what can they do? Can they be stable? Can they be flexible? Strength is something given more by bones, and so yeah, it was a distractor. Okay, what type of ion channel opens to cause the end plate potential? Ligand-gated cation channel. All right, that's all my questions. If you have any questions for me, ask them in the Teacher Helping Students Succeed discussion board. Otherwise, good luck on your test, and I'll see you soon.